Okay, so you may have noticed that our Zoom link has changed, and that's because we have two new organizers with us. So this is a formal welcome to Swati and to Tui Dang, both of whom are assistant professors and have been quite active contributors to the phytochemical community overall and have been regular attendees. Phytochem talks. They have made some really good speaker suggestions over the last several years. Um, with both of them being in the saddle, we've expanded the diversity of speakers to different topics, and looking forward to having exciting talks in the next few sessions. And one of the reasons we got more people on board is because Luke decided to take some time off Phytochem Talks, and he's been a central contributing member of Phytochem Talks. Um, the idea of Phytochem Talks came out from a discussion from an idea I had when I was in the phytochemical section at Botany, and most of the ideas are relegated to the sheep of history. But when I talked to Luke and Lars about this, they really jumped on the idea. And within a couple of weeks, Luke had the first website created for Phytochem Talks. This was our first session back in 2021. We don't have a physical record of these talks, but going back on the Wayback Machine, I could find that our first talk was October 15th. And since then, we have had 65 speakers from 56 institutions, 10 countries, and currently we have Google groups, 323 members on our Google group. And none of these would have happened without Luke contributing quite a lot to Phytochem talks. And there have been instances where he was on vacation, but also joined us from just like Lars also joined us from coffee shops to just start the Minnesota Zoom link so that we could have a nice session. So thank you, Luke, for uh, all the encouragement and contributions and passion that you showed in Phytochem Talks over the last several years. One of the things I personally admire about Luke is this very calm nature, which is also uh, reflected on the front page of his website. Uh, and I would recommend you to go on to his website and see all the exciting research that Luke is involved in. Um, so Luke is currently an assistant professor at Minnesota Duluth. Previously, he was a postdoc and a PGRP fellow in Ed Cahoon's lab at University of Nebraska-Lincoln. And from 2011 2016, he was doing his PhD in chemistry in Reinhard Jetter's lab at UBC. He has many awards to date, and I just took a screenshot from his CV instead of typing them out. But he's been fettered with the Cottrell Scholar Award most recently, which is given to research to faculty, early career faculty who play a significant role not just in research but also in mentoring and teaching, which reflects on the contributions he's had at Minnesota for the undergrads and the student mentors. He's also been an early career awardee of the Phytochemical Society of North America, and he has 38 papers so far and counting. Uh, Initially, Luke's research focused on steroid biosynthesis and lipid biosynthesis, but recently he's also started exploring large language models for better analyzing specialized metabolism. In addition to his research contributions, he's mentored 24 undergraduate and graduate students so far. He's been a reviewer for over 20 journals, if I'm not wrong, and he's been an active member of the Phytochemical Society of North America. If any of you are interested in metabolomics and mass spectrometry, he has also created this integrated bioanalytics course where you can go and look at all the ways by which you can visualize data or analyze data using statistical methods, and there are all the tutorials listed on his website. So I would encourage you to go to his website and check out all these resources. So without further ado, Luke is going to talk about language models to organize decades of article research data. And stop my sharing and pass and let you share.
Thanks, Gaurav.
Okay.
Oh, I don't know if I've ever had such a nice introduction. Thank you. Are the slides looking okay? Okay. Well, I'll begin a little bit. I'll just mention briefly where I am. One of the things I love about Phytochem Talks is that we have people from all over the world, and I think it's fascinating to learn about where those people are tuning in from or making contributions from. So I'll do that here. I live in Duluth, Minnesota. It's right here on the edge of Lake Superior, kind of in the middle of North America, basically. We live in an area with lots of water and beautiful opportunities for going out in nature. There's lots of big, kind of a boreal forest area. Here's our campus. It's a small campus. We're right on the lake itself. We've got the world's largest freshwater sandbar. You can see just behind campus there, which is a huge beach that is a popular place in the summer. My lab is right here in this building here. It's the Center for Chemistry and Advanced Materials Science. And in my lab, we have a number of different research areas. As Gaurav mentioned, we work on plant surface chemistry. This is lipid chemistry, and also involves sterols and triterpenoids. We also work on the phylogenetic distribution of plant chemicals, and we are working to make contributions in that space, trying to understand how the distributions of phytochemicals across different lineages can tell us about evolution or about biochemical pathways. And in support of that aim, we've recently begun this third area that relates to systematizing, or you could think of it as organizing research data with language models. And that's what I'll talk about today. I mentioned that this organization with language models is really supportive of efforts that are working towards analyzing phytochemical data in a phylogenetic context. And I really want to sort of briefly want to touch on that. So the premise here is that analyzing phytochemical data in a phylogenetic context, and this could be enzyme activity data or plant insect or plant pathogen interaction data, or what I will call in this talk plant chemical occurrence data, which means data regarding which particular species of plants are producing which particular chemicals. Analyzing any of those data types can tell us about evolution. It can help with pathway elucidation and more. Here's just one example, sort of a famous one looking at tyrosine enzyme activities, and it helped with the discovery of tyrosine synthesis enzymes that are not feedback inhibited. And this has led to a variety of synthetic biology applications. For this type of work to proceed, though, it requires lots of data. And fortunately, especially for plant chemical occurrence data, there are lots of data points in the literature. For many decades, scientists have been reporting from all over the world, this plant species produces this chemical, and a different plant species produces a second plant chemical, and so forth. But these data are not always systematically organized. And there have been some very substantial efforts to organize it, but databases sort of by definition are not necessarily up to date, nor are they comprehensive. And so this is a problem that we want to try to address with a new technology that's emerging. Of course, one way to address it would be to read many papers and tabulate data manually, but this is slow. So the alternative solution that we're envisioning is using language models to extract and tabulate phytochemical occurrence data. And our sort of overarching question here is whether this task can be completed with accuracy, or at what level of accuracy can this task be completed by language models. I want to talk a little bit about how language models work because I think it's fascinating, and it also provides a bit of maybe a bit of like uncovering what's inside the black box, showing us that language models are not some type of magic, nor are they like some type of sentience at this stage. So the concept here behind language models is that we take a word. Here you can see the word see. Is my mouse visible to you? Yes, okay, thank you. So you can see the word sea and you can see the word here ocean. And we're encoding each of those words with a series of numbers. And those numbers are represented by this heat bar here with low numbers close to zero, having light colors, and sort of the more pink colors being, like zero point 0.8 and zero point 0.9 and so forth. So sea is represented with a series of numbers. And ocean is represented with a series of numbers. And because those concepts are related, the numerical vectors that represent them are similar. Same for football and soccer. You can see they have distinct but somewhat related numerical vectors. Same for I and we. So we can encode words as these numerical vectors. This is the first idea. The next idea is that we could modify those encodings according to the environment in which the word occurs. And so this word interest, it could have two different meanings. I could say I'm interested in something, or I could be referring to an interest rate, the financial meaning of the word. The numerical encoding for that word will be modified by the words around it. And so if the words around it are the bank's interest rate rises, the numerical encoding for that instance of interest will be different than the numerical encoding for the same word, interest, in this first sentence where it's I have no interest in hearing about something. So these two encodings are modified. You can see in the lower one here, it's the other usage of the word interest, the financial usage. So these two encodings for the same word are modified slightly by their context. And this gives the model the ability to sort of quote unquote understand the general context or meaning of a sentence in more detail than just its word components. Yeah. So now through doing this encoding, we've generated, or the model has generated, I suppose, a series of numerical vectors that kind of capture the meaning of something. And it would be possible then to have the model give some type of output as a response to the input sentence. And that output could be a number, it could be some type of summary statistic, like the average of those numerical vectors. It could be computing what the next most likely numerical vector in a series would be, like next word prediction. There's all sorts of outcomes that can be generated based on this. But it's this like numerical representation and modification of that representation that are the key technologies underlying transformer-based language models. Okay, if you're curious about this, there's a QR code here to an article from the Financial Times that these graphics come from, and it's a great article that expands on these ideas more. At any rate, there's, as I mentioned, many different model types. There are some models that we refer to as embedding models, and these are models that take complex inputs like words, or it could be images, and they run that initial conversion that I mentioned, and they convert the word or the image into a series of numbers that are then compatible with downstream statistical techniques, machine learning, and so forth. There are also chat, or what are sometimes referred to as completion models. And these are models to which we give some type of question or prompt, we often call it, and it responds with an answer. And that could be a single word, a series of words. If you used Gemini or any of ChatGPT or the large sort of frontier language models, they belong to this kind of category. There's also classification models which label an input content based on a series of predefined categories. I'll talk about that a little bit later. And there are so-called multimodal models, which can accept multiple types of inputs like text or images or a combination of the two and generate some output based on the combined input of the several modalities. In this case, though, what I would like to touch on is that though there are many applications we could envision of using these models in phytochemistry, I think one important category is going to be the extraction of data from research publications that we as a community have already created and organizing those data in public repositories so that we can use the results that we've collected over decades as best as we can. And the example that I'll talk about today is mapping phytochemicals onto phylogenies. This is the amassing of chemical occurrence data and putting it into a phylogenetic context, as I described earlier on. So the general premise here and the way we began, this was several years ago now, was to start by getting references to compounds of interest from a literature database. And we chose SciFinder. If folks aren't familiar with SciFinder, I encourage you to check it out. It's an incredible research tool, especially when it comes to chemistry, including plant chemistry. It's not just for like synthetic organic chemists. If you haven't played with it, it's pretty powerful. It's like a search engine. It can do structure searches. You can do searches based on plant taxa. It's quite powerful, and I suspect your institution, if you work at a research institution, has a subscription. So we use that to retrieve abstracts that referred to at least one of 70 different tyrosine derived compounds. And then we grabbed from, at the time, World Flora Online, a list of different plant species, their accepted species names. And then we used just simple text matching. So, like a Python script that did text matching, and it's looking for the names of plant species in these abstracts. And if it finds the name of a plant species in that abstract, it looks to see if our chemical names of interest also occur in that same abstract, and it populates a list of candidate associations or candidate occurrences. It says, hey, the name of this chemical and the name of this plant co-occur in this abstract. And it could be that that abstract is describing evidence for that plant making that specific chemical. That might not be the case. It could just be that those two terms, the plant and the chemical, co-occur in the abstract, and there isn't actually any such evidence. But this list of candidates means that there might be such evidence there. And so then our goal was to evaluate those occurrences, and we did it manually, and then we asked the language models to do the same. So as I mentioned, we did this for 70 different tyrosine-derived metabolites. This was in collaboration with Hiroshi Maeda's lab at UW. This was like a COVID at-home project. So our two labs together, we read 2,500 abstracts and evaluated that many candidate occurrences. And we populated this map here. So this is a map where we manually read many abstracts and noted which chemical compounds, which are here on the y-axis, if you will, which chemical compounds occur in which lineages. And you can see there are some positive controls built into this. Here we have amyloidaceae alkaloids occurring in the asparagales, as we would expect, beta-cyanins and beta-xanthins in the caryophyllales, which is well documented. And there are some new things that were discovered as well. These clusters of dopamine compounds in particular lineages were especially interesting to Hiroshi's lab, who studies tyrosine metabolism, and they're conducting follow-up studies on those lineages now. My lab's interest, however, was not in just generating testable hypotheses from this mapping, but in determining whether a language model or the extent to which a language model could have generated that same map from the abstracts. And so an undergraduate student in my lab, Braden Johnson, was interested in this, and he took that spreadsheet of candidate compound occurrences, and he passed those entries one by one to a language model, and he asked the question, here is an abstract. Does this abstract provide experimental evidence for this compound being produced by this species? Yes or no? And then the model would respond with yes or no. And we compared the model's response against our manual evaluations of that same question. And so the model can give sort of, this is a confusion matrix. It can give sort of false negatives, false positives, true positives. And this was years ago. This was in 2022 or it would have been 2023. And so actually these models are not available anymore. But at the time we tested 5 models of increasing size. And that size term refers to the number of parameters in the model. You can sort of think of it as the complexity of the model, or another way to think about it is how big a computer you need to run the model. And what we saw for false negatives and false positives, which I'll explain explicitly what those are in a second, that's shown here. What we found was that the larger models, the sort of like more sophisticated ones, quote unquote, have a smaller false positive rate. That means there are fewer instances of them declaring that there is indeed evidence for a particular plant making a particular chemical in that abstract, when in fact there is not, right? The false positive would be when the model says, yes, that chemical is in that plant, when the manual inspection did not find that same thing. So there's differences here in model size with regard to that false positive rate. That false positive rate, however, was accompanied by a higher false negative rate in those same larger models, meaning that there are true occurrences being reported in those abstracts that the models are missing, that they are not reporting as true. So one way to think about it, at the risk of anthropomorphizing, is that these sort of larger, more sophisticated models are more skeptical, but they also have higher false negative rates. So they're not making as much stuff up as the older models, but they are missing more true associations. There's more details on that in this paper here if you're curious. So it seemed to us that model size at that time mattered quite a bit. And we became curious about what other types of parameters should we be exploring. As you know, model size, of course, is one, but there's many others. So I'll talk about a couple others quickly. One that you may have heard of is called prompt engineering. And the prompt is the specific instructions given to the model regarding the task it is to perform. And so in the previous example, I phrased the instructions verbally to you when I was explaining the experiment. The instructions that I explained were, here is an abstract. Does the abstract provide evidence for this plant making this particular chemical? Yes or no? And so that phrase that I just uttered, that was an example prompt that we could give to the model. But that same set of instructions could be phrased in many different ways. I could have said, true or false, does this abstract support the idea that this plant is found in this chemical? And which is the same instruction, but it's not the same phrasing that I used the first time I told you about I described that task to you. And it turns out, people have reported in literature that the exact phrasing of the prompt has an impact on the performance of a model in a particular task, which seems sort of surprising. But we wanted to test that notion in our context, in this context of mining phytochemical data. And so we used a language model to generate 50 different 5-sentence prompts that explain the same tasks. We manually inspected these prompts, and they all issue the same instructions, just in different ways. Some use a list, some use a markdown format, some use phrasing in a particular way or a different way, and so forth. And we tested essentially the performance. Here we're measuring the percent of correct identifications of, yes, this is a true occurrence, or no, it's not a true occurrence, across these different prompts. And you can see there's a distribution. Many of them perform sort of with an 80% accuracy, but some perform not as well, and some perform better, more like 90% accuracy across our test cases. We used an embedding model. This is a model that converts text into numbers. We used an embedding model to embed each of those 50 prompts. And then we ran PCA on that. So this is clustering the prompts according to the syntactic meaning of the prompt. And then we clustered them somewhat naively here. But what I want you to see is that the clusters, their performance is coded in the colors here, and the performance is correlated within the clusters. In other words, here in cluster 3, the performance is significantly lower. So the performance of these prompts is significantly lower than the performance of these prompts over here in cluster 4, here at cluster B, or excuse me, ANOVA group B. And what this means is that prompt specifics on the word level can matter in getting a language model to perform a task with as high accuracy as possible. So this is another parameter that needs to be explored when setting up a language model to do a particular task. We've experimented with a couple other things. Braden wrote a script to do what's called retrieval augmented generation. This is where, in addition to providing the abstract, snippets of the full text are also provided to the model based on the relevancy of those snippets to the question at hand. We also experimented with a type of prompt engineering that includes particular roles for the model, where we would tell it, you are an AI classifier, or you are an expert in scientific literature analysis, or whatever. And it turns out that providing a role, strangely enough, to the model as part of the system prompt really increases its performance. In our test case, it lowered the false negative rate and increased the true positive rate. By including snippets of the full text, the performance was increased slightly, like a detectable and statistically significant amount, but it was relatively small. But providing these roles here seem to have a substantial impact, which has been reported in other areas where people are studying language model usage and programming. Okay, so providing some context and stating a role, improved the performance in our case. I want to take a step back for a second and talk about sort of language models as they are. So those studies that we did, that was a few years ago. And I want to talk a little bit about how language models are today, especially these frontier large models. So these large general purpose models, these are the ones that, if you go to chatgpt.com or if your institution has a subscription to Gemini or whatever it might be, these are what we refer to as large general purpose, or sometimes they're called frontier models. These models are massive, meaning they have many billions of parameters, or actually for some of them that may be trillions, we don't actually know. And their energy requirements are substantial. And this has been in the news lately. So here's from the last couple of months. Here's a couple of articles describing, or at least attempting to describe, how much energy usage or how much energy is required by these models. And the long and the short of it is that to run the models, these large frontier ones, at the scale that they're currently being run, we don't know how much energy they take to run. That information is not being disclosed. Academics have attempted to estimate based on projections. And so some estimates are here on the left. The first estimate I'm presenting to you is the amount of energy it may take to create one of these models from scratch. And maybe it requires somewhere around as much electricity as 400 US homes would use in a year to create one of these models. And that once the model is created, it also requires energy to run it. And ChatGPT and other large model providers are receiving millions of queries per day. And this is where there's quite a bit of energy being consumed. So the daily operational resource usage for ChatGPT, this again is an estimate. This is not official, but these are the estimates. The electricity usage estimates are comparable to what is used by 35,000 US homes daily. And to cool the servers that are running these models, the freshwater usage in terms of evaporation would match the daily drinking needs of around 3,000 people. And the carbon emissions would require a Chicago-sized forest to offset. And so this is just for one model provider. For reference, this is somewhat comparable to the energy required for Bitcoin mining, which is generally understood to be highly energy intensive. This is, I guess, another point of comparison is like this is somewhere on the order of 500 times the energy required, energy requirements of like a large high performance computing cluster. Another part of this discussion that I think is, I'm not trying to make any arguments here, but I think it's interesting. There's another analysis here last year, and their quote in this analysis is, while large language models have substantial environmental impacts, their relative impacts can be dramatically lower than human labor in the US for the same output. Excuse me. So for a human to do the same thing, the relative impacts might be lower for the model, which I guess is a part of this discussion as well. At any rate, the reason that I'm bringing all this up is because if we're going to use these models to organize our phytochemical research data, we should be aware of these problems and of the environmental impact of these models. We are not the only people, of course, like concerned about environmental impact. People in other sectors are concerned about this. And one of the consequences of the development of these large general purpose language models has been the spurring of the development of so-called small models. And I have them listed up here in the title as small local language models. These are models that function very similarly to the large frontier models, except that they can be run on your laptop. They can be run on local hardware. And there's a hub called Hugging Face. Huggingface.co is sort of the hub for this open source small language model development. And this is a screenshot I took earlier this week. You can see there are millions of these models on here. This is because individuals can modify them and upload variants and retrain them and upload variants and so forth. These models are often built for specific tasks. So you can see in this slide here some of the tasks that are listed: text generation, text to speech, text to video, and so on. Generally speaking, these models can be run on local consumer hardware like your laptop or a computer that you may have in your laboratory. And these have much smaller environmental impacts. Many of them, for example, have environmental impacts similar to watching a Netflix stream or to running a web search. We became intrigued by these small models because these to us seem like perhaps a way to conduct our phytochemical data organization without have it, or in a less impactful way, right? A less impactful alternative to some of the large frontier models I was describing a few moments ago. And so the next task that we undertook was to evaluate some of these small models in the same way that we had evaluated the larger models. So I'll briefly present some of those results. The first task that we attempted to complete with these small models was what I'll call a filtering task. And the idea here was to take, again, SciFinder, the search engine, search for 6 different triterpenoid structures in this case. We retrieved about 100 abstracts. And then we wanted to filter these abstracts for those that report the presence of a particular compound in a particular plant versus those abstracts that do not report that. And we did this, so we had a set of humans did this. Some folks in my lab took 1,000 abstracts, read them, marked them as either important, meaning they report a chemical occurrence, or not important. And then we had the small language models do the same. And what we found, so these small language models, the way that they work, or the way that we set them up, was to assign a score to the paper from 0 to 1. 1 meaning high certainty that it's reporting the occurrence of a compound in a particular species, 0 meaning it does not report that. And so here you can see the data for the abstracts referring to one particular compound. for those articles that we as humans labeled, yes, they're reporting the occurrence of a particular chemical compound in a particular species, those were generally assigned a high score by the small language model, close to 1. For those that we found did not report an occurrence, they were assigned generally lower scores. And for somewhere, or excuse me, for those articles where it was unclear, where it was implied that they found a particular chemical in a particular species, but did not explicitly state so in the abstract, those were assigned somewhat middling scores by the model. And this holds across a variety of compounds, although I acknowledge these are just a handful of the vast phytochemical diversity. But nevertheless, we identified a threshold in this particular case, a value of 0.89 that we could use as a filter to filter for articles that seem likely to report chemical occurrences versus those that might not. And if we use a threshold-based filtering approach here, this allowed us to set up a system where we could retain among these 1,000 abstracts, 92% of those that indeed reported occurrences and eliminate about 55% that did not report occurrences. So this is a useful filter in this context. Not perfect, but useful, I think. The next task we asked the small language models to do was the same task that we had asked the large models to do. This is to look at an abstract and evaluate whether it is providing experimental support for the presence of a particular compound in a particular plant species. And we did this using sort of the top system prompt from that previous work. And we're running this on local hardware. This is a small GPU computer. For those of you familiar with nanopore, we have a nanopore sequencer in my lab called a GridION. It has a GPU in it for doing base calling, and we use that system to do this work. The point being, we ran this like locally, right? And don't have like an evaporative cooling system that's like churning through aquifers. Okay. This should be filled in here. So we presented it with 400, no, that's not true, with 500 candidate occurrences. Does it say in the next slide? Thought I had worked that out. Yeah, 500. 500 candidate occurrences. Of those 500, so this is 500 abstracts and questions whether that abstract supports the presence of a particular chemical in a particular species. 500 instances of that. 70% of them, the correct answer was no. The abstract does not support that. 30% of those 500, the correct answer is yes. The abstract does support the presence of that chemical in that species. So we asked the language model to evaluate those claims. And for the 70% where the correct answer was no, it got this particular model. This model is called Quen 2.532B Instruct. It correctly labeled 67% of that 70%, where that bar is back there. It correctly labeled them as no, the abstract does not support the occurrence. And of the roughly 30% where the correct answer was yes, it correctly labeled 26% of them, so most of them. So again, this means that we have somewhere around like an 8%, 9% error rate. And this varied, but was somewhat consistent across a variety of different small language models. So these are models from a variety of different model providers down here at the bottom. These are all ones that we could run locally, and you can see there's some variation in how accurately they perform. Generally speaking, they perform reasonably well. When the models get quite small, like 1 billion parameters or 500 million parameters, the performance begins to drop off. If you're curious, you can find this study online. This is published now, and you can read about the speed with which these models operate and some of the other parameters that are associated with them. So at this point, we had established that we believe these small local language models, with regard to their accuracy in these specific tasks that we tested, is pretty comparable to large models. They can have around a 10% error rate. These small language models, in contrast, are accessible, meaning you can download and inspect them. They're democratized. They can be edited by the user. They keep your data stored locally. Your data are not sent to remote servers. And they have much smaller environmental impacts on the order of web searches or playing a video game or base calling your nanopore sequencing data. Okay. We wanted to challenge these models with a third task. And this is the third task that we worked on here, and I'm calling it untargeted data extraction. And this is a task where, rather than presenting the model with an abstract and saying, is this chemical found in this species, we present the model with the abstract and simply say which plants are producing which chemicals. and it should list all of the occurrences. So a correct answer for a particular abstract might look like this, where the answer says Catharanthus roseus is making vinblastine and vincristine, and Mitragyna speciosa is making mitragynine. And note that there's a particular syntax here that has to be used. There are these curly braces, these square brackets and quotes and so forth, and this is so that downstream Python scripts can understand the output of the models. So we wanted to first assess whether the models could quickly output a correctly structured response so that it would be compatible with downstream steps. We again took SciFinder, ran the search, got 1,000 abstracts. We manually evaluated 100 of them and wrote out what the correct response is for which species are making which chemicals, and then asked a variety of models to do the same. And again, the first thing that we evaluated was simply, is their output format correct? Not, is the phytochemistry correct? Simply, is the output format going to be parsable by downstream steps? That's what I'm showing you here. In the top, we're looking at the percentage of valid responses, meaning the percentage of correctly formatted outputs for a variety of different models. There's 8 different models and 11 different prompts that we used. And you can see that both the prompt and the model have an effect on the percent of valid responses, but there are several models for which almost all prompts elicited correctly formatted outputs. That's what the 100s mean here. We also looked at how quickly the models could do this, and this is in 1,000 references per hour. So several of the models, like 5.4 mini instruct and Quen 2.57b instruct, they were able to process around 1,000 articles per hour on our computing system. So now I'm going to tell you about this sort of second assessment here, which is arguably the more important one. This is whether the outputs from those models are accurate. And we actually not only care about accuracy, but we also care about comprehensivity. If an abstract is reporting 10 occurrences, the presence of 10 specific chemicals in one or more species, we want the model to output all 10 of those and to do so accurately. And not just to output 5 accurately and skip 5. We want to do all of them and do it accurately. And so I'm going to talk about how these data are represented here, and then I'm going to present you the results. This is just sort of like a mock representation to explain how these bar charts work. The bar chart has a positive y space and a negative y space. In the positive y space, the top bar here indicates the number of correct occurrences initially reported by the model. So the number of correctly reported occurrences across all the abstracts. And across all the abstracts we tested, there are around 400 correct occurrences, 400 true occurrences that were extracted by the humans. In the negative y space is the number of incorrect occurrences initially reported by the model. So this would be if the model had said Catharanthus roseus makes vinblastine, vincristine, and mitragynine, that entry of mitragynine there at the end would be an incorrect response and would be tabulated here in the negative y bar. Okay. The next layer of bar represents the results after we performed a filtering step on the initial results. We took the initially reported occurrences and filtered out the occurrences that we're talking about a chemical whose name was not recorded in PubMed. And so, for example, this would be if the model had said Catharanthus roseus produces zymogen, which is incorrect, right? Zymogen is the name of like a molecular biology company, and it seems like that somehow got confused in the abstract, and the model thinks that zymogen is a chemical that's being produced by the plant, which is obviously incorrect. So by filtering out those reported occurrences that don't have a chemical name in PubChem, we managed to reduce the number of incorrect occurrences, generally speaking, by a greater percentage than the amount by which the correct occurrences were reduced. And yes, this means that that PubChem is not comprehensive. There are some true chemicals that don't have a name in PubChem. We also then performed a final filtering step, and this is where we only accepted those occurrences that were reported independently by 2 models that were asked to perform this extraction, this untargeted extraction. And so if 2 models independently reported the same occurrence, we allowed those to enter the final data set, whereas if only 1 model reported it, it was eliminated. And generally, again, the impact on the incorrect answers was greater than the impact on the correct answers. Okay. So here are some of the data. Here you can see we've got an initial set where this particular model, there are 3 bars here because we tried it with 3 different prompts. This is where a particular model is reporting around 300 correct answers initially and around 175 or 150 incorrect answers. But after the filtering, we winnow this down to around 200 correct answers out of the 400 total that the humans identified and 25 incorrect answers. This is for abstracts that are reporting, on a single species. That's why it says 1 SPP up here. This picture changes when we apply this approach to abstracts that are talking about more than one species, which you could think of as potentially a more confusing abstract. When there are abstracts reporting on 2, 3, or especially 4 or more than 4 species, many more incorrect responses are given by the model, and many fewer correct responses are given by the model. And so here we created a cutoff where we didn't trust any of the results provided by models when there were more than 3 species present in the abstract. And if we look at just those responses provided when we are only allowing the model to process abstracts that report on 1 or 2 species, we see this type of average performance here, where we've got around 225 correct answers out of the 400 and 25 incorrect answers. And this translates into roughly about 55% yield, or recall is another way to name that statistic. And of all the occurrences of all these 225, or excuse me, 250 total, roughly 90% were accurate, which would be a precision statistic, meaning that 10% of the model extracted occurrences were not technically accurate. And this was usually a situation like this, where there's 2 species being reported and one chemical is misattributed from one species to another or to both when it should only be attributed to one. I'm briefly going to talk about one last thing and then I'm going to wrap up. I mentioned earlier that there are these things called multimodal language models where they can accept images as inputs. So we've been doing a bit of experimenting with whether these models can take a table like this and translate it into a sort of a parsed CSV format like this that we could use for sort of data aggregation. And sort of to make a long story short here, we collected 13 tables describing the abundances of phytosterols in different seed oils. These are across the 13 tables, they describe 67 species. So many different tables describe the same species over like repeat species across the tables. And what we had to do was in the end, in order to obtain like high accuracy and in order to make the outputs from the models easy to parse in downstream steps, was that we created versions of these tables, we had to create versions of these tables that were redacted, meaning that like some of these distracting features in the tables needed to be removed from the tables, like erased, manually covered up so that the model would focus on the portions of the tables that we, the scientists, knew contained the data we wanted extracted. So this percentage column here, we would have to cover up. You can see down here, this is a summary statistic row. This is total phytosterols at the bottom, which is the sum of these other rows. That would have to be covered up. Here where it says apricot kernels, we would need to take the actual species name from the text and replace it here in the image in order for these values to be attributed to the correct species as opposed to just apricot kernels. So these tables required manual sort of processing in order for the model to be able to process them in a way that we found to be useful in a straightforward way. So this process isn't perfect yet. It's meaning that it's going to be difficult to completely automate table extraction. It requires human intervention, which I would argue is actually a pretty good thing. In manually annotating these tables and cleaning them up for extraction, we noticed a variety of important aspects of the data that are in these tables that are important to be aware of as a subject matter expert that is the one that's actually conducting the research. At any rate, once we had manually annotated these tables, we extracted 400 compound abundance data points from them in about 2 minutes with a vision language model. It had about 98% accuracy. It messed up very few digits. And we created this distribution map from them. So here we've got the different species in the tables, the different phytosterols, and you can see that in this lineage here, something has happened such that the ratio of stigmosterol to beta-cytosterol has been altered in a statistically significant way. And so this data amalgamation process that we did here maybe points to this lineage, the APACE, as a lineage to look into if we want to understand this particular chemical phenomenon. At any rate, our conclusion here that image models they have some potential in structured data transcription, but we need to be careful. There's a lot of room for improvement, and the conditions matter. I'll sort of wrap up here. We think that language models have potential to convert sort of disparate unstructured data into structured data that can generate testable hypotheses, as I showed you with our tyrosine derivatives early on and with the phytosterols now. We think that many optimizations are possible, as we've shown from a sort of analytical standpoint. These small language models are really critical because I think they're going to allow us to amass our phytochemical data into large usable forms without having an outsized environmental footprint, and used carefully. And perhaps in a few years, we'll be able to do quantitative data as well on a more rapid scale than we currently can. Currently, it can be done, but it's a little bit slow. It requires some manual intervention. Another observation here is that LLM optimization is highly task specific. Strategies that work for one task may not work for another. And so as we develop language model pipelines to help us organize our data, we need to be conducting empirical tests in which humans are in the loop and manually performing the tests at some scale alongside the model to evaluate the performance, because we cannot take performance for granted. All right. I've reached the end here. I want to thank the folks that did the work. This is mainly done by Braden and Bree, who are working on language models in my lab, but we also have folks working on surface chemistry and the evolution of phytochemicals across the lineage Kalanchoe. Special thanks to these folks here that have either collaborated with us or given us support. And thanks to the RCSA and the USDA for supporting the work in my lab. And I'll take any questions that you have at this time.
Thank you, Luke. We have time for questions now. If you want to raise your hand and speak up, you're free to do that, or you can put your questions in the chat. I can start with one. So you talked about small language models. Have you compared the predictions or performance with the lighter models of the large language models like Nano, like GPT-5 has a Nano model or Gemini has a Flash model, which are less parameters?
That's a good question. No, we haven't benchmarked any of the like small versions of the frontier models. Um, yeah, I mean, the short answer is no. I imagine they would perform.
Oh,
I'm not sure what happened there. Can you hear me all right?
yes.
Okay, so we were talking about the small versions of the frontier models, and the short answer is no, but I suspect they would perform pretty well.
Okay. Aaron?
Hi, I had a question about how you sort of chose your data to train the models on. I know like SciFinder is like a really good tool for that, but I've also seen some like publications or with like abstracts that even have like misinterpreted some results in them. So how do you ensure that your models are training on good data so that you have like good results from them and it's not misattributing anything?
Yeah. So I guess maybe I'll say two things here. One, to be clear, like we're not training these models, so we're using models that other people have created or trained themselves. The work that I presented is we're simply evaluating how well those models can perform a task that we as humans can perform that relates to phytochemistry. But you raise an important point, which is that the abstracts, which is what we're basing sort of this like evaluation on, may not be clear, or they may be wrong. They may be misdocumenting what's actually in the full text. Or if we as humans were to read the full text and as trained scientists, we may realize like, oh, I don't think that what they have written here is actually correct, whether or not that made it accurately into the abstract or not. This is something that I mean, we haven't systematically gone and looked at the number of abstracts in our data sets that are not consistent with the underlying full texts. But we have found that a substantial problem, and maybe I think a more common problem, is that abstracts can be vague and non-specific. And so the full text may contain more illuminating information that may give a solid answer that a trained scientist can easily interpret as to whether or not a particular plant is making a particular compound. But that information might not be expressed well in the abstract, and that can lead to confusing outputs from a particular model, which is a challenge. Does that answer your question, Aaron?
Awesome. Yeah, thank you. I've just seen like so many like I think there's like this cyclical thing of like abstracts using like AI, and I've seen some wrong results with that. So I wonder like how that affects it
Mm.
too.
Yeah. Yeah.
Okay. So we'll go to Alex, then Lars, and there's then a question in the chat.
Thanks, Luke. Great talk. And I may have asked this question to you before in person, and this is related to what Aaron's question was. So it's it's maybe not getting at the accuracy, but when you had the human um, abstract checkers go through, if there was a report, say, in a supplementary figure to the paper, was that flagged or was that totally ignored, even by the human observers?
In some of our manually curated data sets that I think of them as like benchmarking data sets, some of those we have columns where we have noted where the abstract is unclear, but it seems that the full text or supplement contains the true answer or like will contain like the details necessary to clarify the situation. And I think that was your question. So
Yeah.
like we
And I
have
think you had a student working on like a language model that could then recall that
Oh,
information
right.
too,
Yes.
right?
So
Well,
yes.
I guess it's two-parted that
Yeah, I
question.
see. Yeah, correct. I remember your question now from when we were in person.
Yeah.
So, yes, basically the problem that we run into so at that time, yes, we were flagging those where it's like, okay, we need the full text or the supplement to get the real answer, and we'll have somebody work on that. And what the problem that we ran into is that many full texts and supplements are copyrighted material, and it's prohibited to upload them. I would argue it's like unethical to upload them to large language models on remote servers that are potentially being further trained on this copyrighted material. And so this is what one of the things among, in addition to like the environmental impacts that initially got us interested in the small language models, because not only can we run them without consuming as many resources, but we can also then we can give them copyrighted material because we're not uploading them anywhere. We're processing it as if we were to open the you could think of it as being analogous to opening the full text in Adobe and doing like a control F for whatever you want to find, right? It's a more sophisticated version of that essentially when it's a small local language model. And now that we've benchmarked the small local language models, I think we're in a better position to answer the original question of can we take these ones that have been flagged as needing full text or supplement and evaluating what type of gains we get when we use those. So I think that's a next step that we thought we were in a position to address, and now I think we actually are in a position to address, if that makes sense.
That's great news. I'll look forward to seeing that.
Thanks, Alex.
Yeah, I guess I had a very similar question, like in terms of the quality of abstract, and I was wondering if you did any comparisons between like supplying like the model with like a full text versus like the abstracts. And then second question is basically, can we derive like some sort of rules for writing good abstracts from what your models are like kind of finding? Because then going forward, we should write better abstracts to make this easier, right?
Yeah. Yeah. It really, okay, yeah, I can say a few things. So for like we've done one experiment where we tested what types of gains in accuracy or really like comprehensivity, I think is the main one, the main metric in this context when full texts are provided. And there are like gains, but really that the amount that's gained there is very dependent on the benchmark data set. If we have a benchmark data set that's comprised of abstracts where all the abstracts contain the relevant information, then providing the full text isn't going to appear to have that much of a gain. But if we have a benchmark data set where lots of the important information is in the full text, then it will appear as if providing lots of full text provides like lots of gains. So it really depends on what the input material is. And then, sorry, what was the other? I had something else
The other
interesting.
question is like, can derive any rules to like
Oh,
formulate better abstracts, like
yeah.
based on how the models are performing?
So it, you know, if the goal is to write an abstract that will be easy to that will be compatible with extracting chemical occurrence data, then yeah, we could definitely write abstracts that can do that. A lot of abstracts that work really well for that are ones from like kind of like the like organic chemistry side of phytochemistry, where people are doing structural elucidation. They say, we analyzed the bark extract of this plant and here are the compounds that we found. And that's like the premise of the article. Those abstracts are like tailored to this type of thing. But a lot of the articles that we write these days, there's so much more to them than just identifying particular chemicals in particular species. We're talking about interactions, evolution, we're talking about synthetic biology, yields, and all these other things. And we're trying to jam all that into the abstract because all of those are important and interesting points to the reader. And so the, you know, it is possible we could derive some rules for generating abstracts that are easy to extract, but I'm not sure that those abstracts would fulfill their true purposes in the larger scheme of what we're trying to accomplish as plant scientists, if that makes sense.
Yeah.
There's a question in the chat.
Oh, yep. Okay, so the question says, do you think we can, this is from Basanta, do you think we can track evolutionary tree based on those metabolites or chemical compounds in the near future? There is a, I maybe I'll rephrase the question and hopefully I'm understanding it correctly. So is the question like, can we construct an evolutionary tree based on these chemical compositions? Yes. Okay. So this is something. So that type of activity, I at least generally think of as being called like chemotaxonomy, which is using chemical compounds to generate phylogenetic trees. And that is certainly an activity that it is possible to perform. Like we have the technology and understanding on how to do that. But I think that generally, because plant chemicals can evolve repeatedly in different lineages, their chemical profiles are not as correlated with their evolutionary history as are their genomes. And so I think stronger, or I should say, maybe like more accurate phylogenetic trees can be derived from genomic data than from chemical data because there's a stronger connection between like ancestry and lineage and the DNA. That connection is stronger than the connection between ancestry or lineage and the chemical profiles.
Okay. Are there any other questions? We are at the hour past noon here in on Eastern. Oh, Basanta, you had a question?
Yeah, actually, recently I have been encountering with based on the genomic data. Or are you listening to me?
Yeah, I can hear you.
Yeah, based on the genomic data, like there are several gene or the protein or the amino acid sequence. For example, gamma tocopherol methyl transferase in monoterpene indole alkaloid is doing N-methylation, like which is not supposed to do the methylation of N nitrogen compound. So the genomic data it does not looks like accurate because it cleared with the tocopherol methyl like vitamin E synthesis. However, it is doing the monoterpene indole alkaloid. So I was wondering if we can track down the chemical evolution as well as the genomic data. If we can combine both of them, maybe it will give accurate phylogeny or the evolutionary tree. That was the main question.
Mm.
Is that possible in the near future or not? From your
So,
model.
Luke, just before you answer, I just wanted to say that we are past the hour, so I wanted to formally end the presentation, but whoever wants to stay on can stay and we can keep talking. Thank you all for joining.
Thank you, Gaurav. Yeah, thank you all for your attendance. It was great to see you.